Good afternoon, everyone. Thank you all for joining us this afternoon at the AGM of the Royal Society of Queensland. I'd like to acknowledge the traditional owners of the land on which we are meeting today, the land of the Jagera and Turrbal people, and I acknowledge their enduring contribution to knowledge and our culture. We're very pleased today to have two young researchers join us today to give us some presentations. So Emily Smith is researching when and how and where ticks produce alpha-gal in their saliva, a cause of life-threatening allergic reactions to products of non-human mammals and some cancer treatment drugs, and of adverse effects on the cardiovascular system. The prevalence of alpha-gal syndrome has been increasing, likely driven by rapid land use change in peri-urban areas. Now, if anyone was lost after the first sentence, I'm sure Emily's going to explain all of that.
Well, good afternoon, everybody. Thank you so very much for having me. I'm Emily. I'm a current PhD candidate with the King Lab over at the IMB with UQ. Earlier this year, I was honoured to receive a grant from the Society's Research Fund to put towards my work in investigating alpha-gal syndrome in Australia, which is induced by the eastern paralysis tick, Ixodes holocyclus. So, the eastern paralysis tick, or Ixodes holocyclus, is the most medically significant tick in Australia. As you probably already know from its name, its saliva contains potent neurotoxic venom that causes paralysis, and it can be fatal when it gets on a pet. It's also a vector of diseases such as Queensland tick typhus and Q fever. But what I would argue is the most fascinating thing about this tick is its ability to induce an allergy to meat in humans, which is just crazy. So, mammalian meat allergy is also known as alpha-gal syndrome, or AGS. It's a unique condition that can occur following the bite of certain ticks. So in Australia, the tick that induces this syndrome is the paralysis tick. It's an allergic response to a carbohydrate epitope known as galactose alpha-1,3 galactose, or alpha-gal. And alpha-gal is expressed on the cell surfaces and in secretions of virtually all non-primate mammals. So it's not only meat that we can develop an allergy to, but things like dairy, gelatin, mammal-derived medications, and even vaccinations in some cases. There's also been a single case study published earlier this year linking possibly alpha-gal sensitization to coronary artery disease. That's that article down the bottom right there. AGS is further unique among IgE-mediated food allergies in that the onset of the reaction is delayed. So symptoms usually occur between 3 and 6 hours after consuming products with alpha-gal. And these symptoms can range from anything like hives and abdominal discomfort to anaphylaxis and, unfortunately, as has just been confirmed this month, death. Severe cases can indeed be fatal. Okay. So glycosylation, or the adding of carbohydrates to structures, is one of the most common post-translational modifications in eukaryotic organisms. Galactosyl transferase enzymes, or GALT, because I'm not saying that so many times, catalyze the transfer of galactose, the sugar, from donor to acceptor structures via the formation of glycosidic bonds. GALTs have high specificity for what kind of glycosidic bond they can use to link that galactose, and that is what their name comes from. So, for example, a B3 GALT will catalyze a beta-1,3 linkage, B4 GALTs for beta-1,4, and so on and so forth. In competent mammals, the GGTA1 gene transcribes an A3 GALT that links galactose to another galactose by an alpha-1,3 linkage, forming the alpha-gal epitope, which means it's expressed, it's a post-translational modification. It'll be on a lot of the proteins of this organism. And around here in our evolutionary lineage, the gene becomes non-functional due to frame shift mutations, and it's not transcribed in humans, but if it were to be, it would be this truncated, non-functional, just nothing protein. So humans, therefore, we can't produce alpha-gal, which opens up the possibility of us developing reactions against it. But what's the connection to tick bites? So In the early 2000s, researchers in America and Australia stumbled across the link between tick bites and alpha-gal allergy around the same time through serendipitously different methods. So there have been cases in the US of cancer patients having severe allergic reactions to cetuximab, which is a monoclonal antibody produced in mice, and these mice produce the alpha-gal gene. And it's used as a chemotherapy drug. And unfortunately, it took a case of fatality for them to start looking into this further. Over here in Australia, also in 2006, the clinician Dr. Cheryl Van Noonen was seeing several concerning cases of people having reactions to meat. And the only common link she found between all these patients was that they'd all had a history of paralysis tick bites. And she was actually the first one to make the link between tick bites and alpha-gal syndrome. 2009, she publishes her results, and a US researcher, I forget the name, it's not coming to me right now, has actually confirmed that her patients had in their sera IgE specific to alpha-gal. And in 2010 to 2011, a formal link has been published that tick bites can cause this allergy to alpha-gal containing substances. And by 2012, we have a diagnostic blood test available for specific IgE to alpha-gal. AGS has since been reported in every continent where ticks are found. Notable tick species implicated in AGS include the Lone Star tick in the US, which I've just mentioned, the castor bean tick in Europe, and of course, the wonderful paralysis tick here in Australia. Now, AGS has an extremely high burden of disease in Australia, as I'll expand upon soon, but virtually no research has been conducted into the mechanisms by which the paralysis tick can do this. There's been a lot of research on the Lone Star tick and the castor bean tick in Europe and some other Asian ticks, and there are three sort of dominant hypotheses about how it can possibly work. For example, there's a hypothesis where the tick takes a blood meal from a mammal that produces alpha-gal and then bites a human, thus transferring via the midgut. There's another hypothesis where pathogens in the saliva express alpha-gal on their surface, and those are transferred. But recent evidence has shown very strong support for a hypothesis that the tick is actually able to endogenously biosynthesize alpha-gal and decorate its salivary proteins with it. But again, all of that research has been done in overseas ticks, which are quite genetically distant from the paralysis tick in Australia. So we don't really know, and that's my job, is to figure that out. So last year, I interrogated the draft paralysis tick genome using a blast search, and I revealed that the paralysis tick possesses a really wide range of diverse GALT enzymes. It boasts the highest total number of GALTs among the tick species that I've compared it to, so 93 compared to the next highest was 86. And it has the most A4 GALTs and B3 GALTs of other notable tick species implicated in alpha-gal syndrome. It's important to note, by the way, that among every tick genome so far, no A3 GALT has been found. So we think that the tick is able to kind of engineer some of its other GALTs to also catalyze the alpha-1,3 linkage. This has been reported in literature, specifically the B4 GALT7 gene in certain bacteria, and also I. scapularis from the US, the black-legged deer tick. And that's very exciting, and we wanted to sort of test that a little bit more. So we also looked at the expression of this gene through RNA-seq. We used RNA-seq on tick salivary glands. This was done by a past PhD student at the time when looking at ticks feeding on different animals. So we had rat in the black bars and bandicoot in the white bars. The bandicoot is the native host of the tick. So it's interesting to see not only the pattern of regulation of the gene over time, but depending on which organism it's feeding on, it's different. So since the rat is not the native host of the paralysis tick, it seems like it's trying to produce more and more. Maybe it's something to do with the coevolution with the bandicoot. We are not entirely sure. But it kind of suggests that maybe if it bites a human, it could also be doing a similar thing. We aren't a native host. It could be pumping way more in, especially as some people believe it to be a form of molecular mimicry. So flooding the host with these kind of self-antigens or things that look like self-antigens to be like, hey, I'm not here, hide me from your immune system. But obviously we don't have alpha-gal, so we can then develop a reaction to it. Then I also did some qPCR of different tick tissues, and the B4GALT7 gene in particular had very high expression in the salivary glands of ticks that we analyzed right after we pulled them off the host. We also waited about 3 hours after pulling them off the host to analyze them to see if a time delay would cause any downregulation at all. And yes, we do see it downregulate. See if I can get my cursor up here. So here's when we pull it straight off the host and analyze it. And here's about 3 hours after analyzing it. So there is some significant downregulation in there, suggesting that it's linked to feeding and also could be very highly involved in alpha-gal synthesis. And I also used an indirect ELISA to see that not only is alpha-gal itself only present in the salivary glands once feeding is started, but it's not actually in any other organ. And this was a surprise to me because I chose the midgut of the tick as what I thought would be a positive control, because ticks feed on blood from mammals. These are specifically ticks fed on dogs. Dogs have the alpha-gal gene. Dog blood would have alpha-gal on it. So I was thinking, hey, I'll put a midgut in as a positive control and see what it compares to. But in fact, we see no expression of alpha-gal, the carbohydrate, in any other tissue, which lends some doubt to that hypothesis that the transfer of alpha-gal into human skin is by the tick biting something else first and then biting you. It seems to be very quickly metabolized. And so these data together have built a solid foundation on which our understanding of alpha-gal syndrome in Australia can be further built on, and also laid the groundwork for my PhD. Now, as I touched on briefly earlier, Australia has one of the highest burdens of disease of AGS worldwide. But the most recent figure we had for that was from a 2016 governmental inquiry estimating the prevalence of AGS as 112 cases per 100,000 people. And that was specifically in the Northern Beaches area of Sydney, because this was a figure proposed by Dr. Cheryl Van Noonan, who is a clinician in that area. We were able to obtain patient data containing the results of the alpha-gal specific blood test that became available in 2012 to give us very precise and up-to-date insights onto the prevalence of this condition across Australia. In 2024, the areas with the highest rates of positive cases were Pittwater, so that includes that Northern Beaches of Sydney area, that's down this little tiny pocket here. They have 1,145 cases per 100,000 people. Coming in not so close behind, but still very staggeringly high, is Coffs Harbour with 629 cases per 100,000. And up here in Queensland, the Sunshine and Gold Coast hinterlands are pushing 500 cases per 100,000, which really tells us how prevalent this is. And as you can see in this this is a zoomed in map of the East Coast here. It's very prevalent along the coasts, and especially in areas like the coastal hinterlands that are seeing more and more development in these tick endemic areas. I think we can expect to see these numbers go up significantly as the years go by. I've also been using these data to calculate risk ratios, or how at risk certain groups are compared to a reference group for developing AGS. And just a quick go over how to obtain a risk ratio. The risk or exposure rate of your target group gets divided by the risk of your reference group. So then if the number that spits out from that is greater than 1, it means your target group is at more risk, and vice versa for if that number is less than 1.
And vice versa.
So I calculated risk ratios by age group alone, and I used people aged 25 to 34 as a reference group, which is why that's kind of just one here. And we found that every other age group is at less risk relative to 25 to 34-year-olds for developing AGS, which challenges a notion that's been kind of casually proposed, nothing published, that older people tend to be more prone to this condition. In fact, it's the young people that are the most at risk. And I also investigated whether sex has a differential effect on risk. So I calculated these risk ratios of females compared to males aged 25 to 34. And apart from 3 age groups, females seem to be at more risk than males for age groups 15 to 24 all the way up to 84. So what's on the horizon for my project? Well, right now I'm in the process of optimizing and re-optimizing my ELISA protocols so that I can analyze over 60 samples of paralysis tick salivary glands collected from ticks at different feeding stages, so that I can get an idea of how alpha-gal production changes in the glands over the course of feeding. I've also been able to dissect out and fix single glands from ticks from almost every day of the feeding cycle so that I can do some immunofluorescent microscopy on them. So I have my primary antibody specific to alpha-gal and a secondary antibody that's conjugated to FIT-C, so I can get some fluorescence there. And I'm in the process of developing a protocol for that, and I should be able to get started on that very, very soon. I'm also going back over my list of putative holocyclus galts that are likely to be involved in the synthesis of alpha-gal in the salivary glands, because I want to put these into a vector and transfect them into human cells. Human cells obviously don't produce alpha-gal, so this would be a good test to see if in vitro these tick genes actually produce alpha-gal like we think they do. I'm also in talks with some lovely folks at CSIRO Ag and Food at the IMB about the feasibility of doing some glycoprotein, glycolipid analysis of the tick saliva itself, but that's kind of in early stages. And most recently, I've been culturing paralysis tick eggs in the lab in the hopes of being able to generate the first ever paralysis tick cell line for use not only in my research, but also for the wider scientific community to have in their toolkit when embarking on research on this fascinating Australian tick. I'd like to thank you all very much for listening. My hugest thanks to the Royal Society for having me today, and to my team at the King Lab and my supervisors for helping me get to where I am today. Thank you very much. And happy to take questions.
Do we have any questions? Thank you very much.
Of course.
Yes, Julie.
Great talk, thank you. Very interesting. I just recently was tested for, the allergy thing that I'm not allergic.
Oh, good.
Thank God.
Yes.
Very interesting. Do you think the age differences is in part because of exposure? Because you're more likely to have young people out in the bush doing things?
Yeah, well, that's sort of what I'm starting to think, is that, with the rise of bushwalking and stuff like that, well, not the rise of, but more younger people are heading out to the bush and not really knowing how to protect themselves. Again, nothing was published to suggest definitively that older people are more likely, but that was the prevailing kind of thought that I've been talking to. I actually met Cheryl Van Noonen at a conference earlier in the year, and we were sort of discussing that. The theory behind that was, that for some reason, you might not get alpha-gal syndrome after one tick bite, but maybe after a bunch. And if you're older, you will have had more chance to get more tick bites. But this data kind of disproves that. And it's very interesting because this is almost my age group, actually. And there's very little awareness of it. So trying to market awareness to younger people versus more experienced people is definitely going to be something to think about. Thanks for the question. Hi, yes.
Thank you.
Of course.
Most of the people in this room would be bitten by ticks, and many times they're still here.
Yeah.
So what I'm wondering is there a build-up to where you get really bad outcomes? And is there a threshold where you become tick resistant?
Well, that is a really good question, and one that we unfortunately don't have an answer to just yet, because it seems like while every paralysis tick bite carries the risk of developing AGS, not every tick bite is guaranteed to give you it. And there's a lot of factors that we think, it's mostly immune factors that tend to be what's the difference between whether you get it or not, if you're bitten by the same tick, like, whether your immune system recognizes it or not. It's just so up in the air. There's 7 different hypotheses on why. But for the most part, it seems to be that for the most common theory is that multiple tick bites will make you build up a sensitization to alpha-gal. Okay, sorry. So you'll get bitten by a tick. You might not get the disease straight away, but your immune system might get a little bit sensitized to it. And then you get closer and closer and closer until it finally ticks over into clinical symptoms. So for example, one of my colleagues, Steve Barker, he's been bitten dozens of times by paralysis ticks, and he went and got a test for alpha-gal, and he showed me the results. He's like, oh, look, I've got really high, what would be considered clinical alpha-gal syndrome, but he has no symptoms. So it's also possible for people to be very highly sensitized, but also have no symptoms. And we have really no idea why, unfortunately, but it's very fascinating and something that I wish more people were looking into because, especially because I'm only looking into the tick specific side of things, I think it'd be a really interesting avenue for the immunology side of things. So I don't have an exact answer for you right now. It's just a very fascinating field at the moment. Of course.
Yeah, I'm a bit like other people, troubled by the fact that males over 85 were particularly susceptible, apparently, but then the area around Coffs Harbour, there is a range of presumably virulence in the tick populations, which again is very interesting ecological problem to untick, so to speak. Terrible pun, but is there any thinking that you have as to why Coffs Harbour should stand up like that?
Yeah, I mean, hang on, let me get back to the map. Coffs Harbour's around sort of here. Yeah.
Pittwater, of course, is a population. I mean, it presumably these cases are related to the density of population in relation to the population of ticks. So the more people there, the more ticks, the more interactions you get. So Coffs Harbour is a reasonably densely populated area,
Yeah.
I guess. So it's the Sunshine Coast. But Pittwater, you can understand, it's a big area where a lot of people do go to book.
Yeah.
Anyway, that was the question as to whether there is any understanding of the dynamics of the tick populations in spatial.
Yes, well, I haven't pictured the data here, but my aforementioned colleague Steve Barker and one of his students published a paper last year about the effect of the year by year the climate on the tick populations and how early the tick seasons start and how high the peaks are of that. So I would think it would be like an environmental thing. And I've again, because of the population differences in population density of all things, that's why I've kind of standardized by doing a rate per 100,000 people. So Pittwater, for example, the SA3 area, statistical area, only has about 60,000 people. So this is kind of rounded up. It's just a standardized rate. But I would imagine that it's definitely a lot of environmental factors also contributing to, how early in the season the ticks start biting and how many ticks there are per season. But also just the big thing is urbanization of rural areas and the development into, especially in the hinterlands, that used to be completely bush and now it's like a lucrative place to build your multi-million-dollar house. So it's a combination of those factors, I think. And also it just seems that the tick really likes the weather around here, around this East Coast area. So I'd imagine it's mostly environmental combined with just development into rural areas, if that kind of gives a little bit of insight. But again, I'm not an expert in the environmental stuff. That's Steve and Ken.
It would be interesting. It would be interesting to understand whether the ticks had more alpha gal in different places.
Yes, exactly. Yeah. And that's a really good point because the samples that I used in my honours, they were from Lismore. And the samples that I have now are from Pinjarra Hills out in Queensland. So I'm really interested in comparing those once I get the data. Yeah.
Speaking of the experience of one person I know who was bitten by a tick that lodged behind his ear, and he was feeling unwell. He went to a doctor
Recorded.
and discovered, and a couple of days later he was starting to paralysis down the left side of his body. So the tick went to its name. But the question arises is where something was like that for a couple of days, this is more affected by temperature in an environment where it will produce more effect as time goes on because of its access to a blood supply.
Yeah. Well, the thought there is because ticks have multiple stages across their feeding. So they start, the paralysis tick is usually attached for 6 to 7 days. And it's only at about day 4 where they start being really, really fast with their blood meal. And so if I go back to, where are we? Here. So you can see, so I've got unfed day 0, 1, 2, 3, 4, 5, 6. So in the rat, the tick starts producing this gene a lot more on day 4 and 5. So it could just be a factor of, when in the feeding cycle does evolutionarily this thing have an advantage to be expressed more, or where does it start to get more expressed or downregulated? And especially because you can see there's a difference there between the rat and the bandicoot. In the bandicoot, it's lower expression, but it starts on day 3 rather than day 4. We don't have access to ethical concerns with, putting ticks on humans and then doing that. It would be really fascinating to see what the pattern of regulation is when feeding on a human. I think Steve's tried to set up an artificial feeding system, but I don't think it's been very successful. But yeah, it's mostly due to the stage of the feeding and when those certain genes are expressed. And especially because, if memory serves, the paralysis toxin itself isn't expressed until at least day 3, maybe day 4. So it's on there for a couple of days just hanging out. And then it starts kicking up these expressions of these toxins and these molecular mimicries and things like that to try and keep itself hidden, is what we think. I'm not sure if that answers the question because we don't have an answer just as yet, but that's what we are theorizing, if that makes sense. Sort of, yeah. Thank you. Hi.
Emily, most impressive. I come from out west, so we're quite familiar with the little ticks there, which are on the school scrub ticks.
Yeah.
Do not have lunch on the Wiltshire because that's where they seem to look like most of them. They don't suffer from this or what, or we simply don't know.
Doesn't seem to. It seems to be only the paralysis tick in Australia that can cause this disease. And again, I mentioned earlier, it's been observed in every continent where ticks are. That doesn't mean that every tick in every continent will cause it. It's only specific ones. And we don't know what the connection is because, the paralysis tick is not very related to the Lone Star tick. It's not very related to the castor bean tick. They have the genus name in common, like Ixodes. They're in the same family of hard ticks, but that's about where the similarities end. And Steve's actually been campaigning for the paralysis tick to be under a different taxonomic name altogether, Australoxodes rather than Ixodes. So we really don't know what makes it, evolutionarily, what has caused only certain ticks in these continents to be able to produce this in their saliva. I'd be really interested in finding out why, though, because it seems something completely random. But yeah, it only seems, as far as we know, in Australia, that the paralysis tick causes this and no other tick.
It would seem as though this is a very interesting area of inquiry, given the fact that the group is working on trying to help people not get affected by it and die.
Yeah.
Obviously, that's the basis of the group, working on these enzymatic approaches like you do. But it would seem that the ecology of the tick in different areas, this one versus that one, the paralysis tick, and the relationship with the meat eating subsequently, which is the one that really frightens one. It can get potentiated by different tick bites. But again, how do we determine, how does one determine whether the tick I got behind the ear or behind the leg, a knee, happens to be this paralysis tick or not? By the time you get a tick out of your skin, you've squashed it up, but you've not got the opportunity to do any nomenclature on it, any taxonomy on it. So these are sorts of things that, as sufferers, we would like a research group concerned with it to be real well and truly concerned with and answering and looking at it. So you might go back, we can give you a set of questions, I can, to your group and tell them, as a practicing being bitten by ticks around the Coffs Harbour region, I'm very anxious that we get a bit more info in this space.
Exactly. And I'll just really quickly say it is a very big passion of mine that to raise awareness of this condition because it is so difficult to get diagnosed with it. And I would be very interested in looking into that more. So thank you.
Well, you can get in touch with me and I can give you feedback a group from here about it.
Exactly. Hi, yes.
Thank you. Have you got any insight into the density of mammals and marsupials among them that are actually hosts for the tick? And if there's a high density of hosts, is there a relationship between that and the capacity or the vulnerability of humans interacting with that in that sort of environment?
Yeah, I haven't specifically looked into it. I know I mentioned very briefly earlier that what's called the native host of the paralysis tick is the bandicoot, but the paralysis tick is it's very non-specific in the hosts that it seeks, so it can be on basically any mammal in the region.
If you've got high densities of native mammals and marsupial mammals,
yeah,
basically that could be a part of the picture from the other side.
It could indeed, yes.
For the tick to move on to humans.
Yes.
There needs to be an ecological sort of model developed between those two relationships.
Yeah, and especially, sorry.
Sorry, I'm speaking out loud. In some ways it doesn't matter because what matters is that the food link is where I guess, because they are on cows and sheep and whatever else as well, their size, their population size could be dependent on, total.
Yeah. Yeah. And especially because humans are very, very much a host that they will readily feed on at any life cycle stage as well. It's basically I would personally be interested in looking at the populations of native and non-native animals and looking at ticks fed on bandicoots versus something like a dog, which is what I was kind of starting to do in my honours. But especially because humans are a host for any life cycle stage, it makes it quite difficult to definitively say that it's a wildlife population problem because we are just as readily hosts as any other animal to this tick. It doesn't care what it bites as long as it gets a meal, as opposed to something like the cattle tick, which will only bite a cow. It's very specific. It's strange.
And it could be in urban areas with high domestic pets.
Absolutely.
So is there an actual monitoring program on the prevalence of ticks?
I don't believe so. Not that I'm aware of. I know that sometimes vets will literally take the ticks off the animals and put them in a mail envelope and send them to Steve to identify. But that's all I can think of. I don't think there's any official kind of monitoring system for it. It's just based on word of mouth. And when the news starts saying, oh, ticks are in season, it's just because more vets have started seeing them and there's no official kind of...
Is there some scope for science project?
Possibly. I'd be very interested.
The folklore is that you get a tick by going unprotected by clothing through grass. And grass is often a long way from any animal. Is there a migratory pattern that goes from a koala or a dog or a sheep, many of which are a long way from where I've been picking up ticks in and around Coffs Harbour?
Yeah.
The general ecology of this particular tick would seem to be a worthy area of investigation.
Yes, I know that when they're feeding on bandicoots, they tend to live in the burrows or the dens of the marsupial, but that's just on the bandicoot. For when we collected these unfed ticks, we collected them what's called by the flagging method, which is where you just go in the bush, have a piece of fabric and just go try and get them. But when the ticks are, especially the adult ticks, when they're looking for their first meal after molting, they exhibit what's called questing behavior, which is where they have their front legs up and they're just kind of looking for a host because they have sensory organs in there, and they move quite quickly as well. So they can cover relatively high distances, I assume, but I don't think, to my knowledge, there has been any research into that kind of behavior. All it is just, observations. Yeah, so far.
I guess we need a few more deaths in order to get money into the system of ecology.
It is very tragic that has to be, yeah. Hi.
A query.
Yes.
Is there best practice in how to get rid of the tick? Like in terms of snake bites, we started off with slicing with a razor or a knife. that's why it's not a good idea.
Yeah.
I suspect the same that ticks my usual thing is just pull them off.
Yes.
And leave it behind and wonder why it isn't there.
Yeah. Well, yeah, well, a lot of research articles, or not a lot of research articles, a lot of news articles will say the best way to remove a tick is by freezing it off or by like getting it at a certain angle and like, or just like leave it on until someone can get it off. But like, that's because there's kind of this notion that like, if you remove the tick with force, it will inject more toxin into you. But in the grand scheme of things, a tick is only injecting like 0.001 microliter of saliva at that point. Like my colleagues at Cyro Ag and Food that I was mentioning in my last slide, they had done salivation experiments on these ticks and they got infinitesimally small amounts even after putting on a substance that makes them salivate. So according to Steve, who's like our resident expert in ticks, just get them off as soon as possible, just because the amount that it can then inject into you when it's on for longer is going to be more than if you just pull it off straight away, if that makes sense.
Maybe as you recall, conversation is the practical basis of the.
Yes, I think Steve actually was either he was in the news, like the television news, or on a ABC radio. He was actually talking about his findings with that as well. I hope he's going to publish a paper about it soon because I would love to get that out. Oh yes.
The latest recommended practice is to freeze the tick to have it fall off naturally. Otherwise, in squeezing, there's the risk of the foreparts off, and they'll stay embedded and continue to intoxicate.
Yeah, I'm pretty sure if you get like a pair of good tweezers and get right as close to where it's in your skin as possible to try and get the mouth parts out as well. Basically, as long as you can get it off as soon as possible, don't leave it on. Yeah, sorry?
Doctors cut them out rather.
Oh, really? Well, there you go. But yeah, obviously the mouth parts will stay in, like if you manage to get off in a bit of a panic and they kind of stay embedded, it's not going to be able to inject any more stuff into you, but it's just a foreign object in your skin that can get infected. So that's when the doctors will be probably have to intervene on that. But if you can kind of finesse the tweezers to like kind of get, if I can go back to my very start where I have the diagram of the tick. So if you can get like this part will be in the skin, absolutely. Usually these parts on the side are splayed out to get it like a good grip. So if you can get the tweezers around that area to pull it out, you should be able to have them not stay in your skin.
Suggestion to twist them as you pull it out. That fouls the mouth cells.
Yeah. Well, it could work. Yes. I came here with one research project. I'm leaving with two.
This one.
Of course.
That's the one from a tick bite, and the lacutamine pipeline got scrub typhus.
Yes.
Does homocycus be a vector for other diseases?
Yeah, I mean, as far as like the most notable ones are Queensland tick typhus and Q fever. I think it can also carry bovine babesiosis, which is like a thing that affects cattle specifically. But in terms of things that specifically affect humans, those are the two main ones that I have seen in literature. Ticks, I think, as a whole, I think ticks as a class of organism carry a vector for more diseases than mosquitoes even are. So I wouldn't be surprised if it can carry more. That's just the human side of it that I've heard is tick typhus and Q fever and some cattle stuff as well. But there's probably a lot more as well in there. The student I did my honors with, well, we had the same supervisor. She was doing a project on endosymbionts in the tick gut, and she found a lot of beneficial ones to the tick, but not specifically disease-causing ones. But I wouldn't be surprised if it can be a vector for more diseases as well. Thank you very much for your questions, everybody.
Thank you very much. Thank Emily for what's been a very